So I'd like to welcome everyone to the first Landesman lecture. Tonight we'll have the first speaker of the first Landesman lecture is Terrie Williams, who's a professor of ecology and evolutionary biology at UC Santa Cruz. I'm Eli Silver, and I'm president of the Emeriti Association. And tonight's lecture is How to Live with a Calculating Cat, Part 2, The Feast of Kings. There will be time for questions after the talk, and at that time I'll remind you, only ask questions when you have the microphone in your hand, because we're recording this. It'll be available on the Emeriti YouTube channel, and there are like 110 people signed up for the Zoom-like thing that we have going. I'd also like to thank Ed and Miriam Landesman for supporting this series. And just say a few words. Ed is Professor Emeritus of Mathematics at UC Santa Cruz, got all his degrees at UCLA, and he has a distinguished record in pure mathematical research, but he also has focused on teaching, research and teaching education, and emphasizing the use of technology. Ed has received a whole series of excellence in teaching awards from UC, from Phi Beta Kappa, from the Mathematical Association of America, and it would take too long to go over all of his awards. And Ed also played a major role in the creation of CalTeach, and he and Miriam continue to support CalTeach. Good. Miriam earned 2 masters from Stanford, one in mathematics and one in teaching. And at UCSC, she taught in both the mathematics and economics departments. And she was recognized with a series of honors for excellence in teaching as well. And more recently, she was the education lead for Smart Skies at NASA. And as a result of that, was honored with several very prestigious awards. And in addition to all that, both Ed and Miriam have been very generous to the Emeriti Association, allowing not only this, but other aspects of things that we do. Now I'd like to call up Dan Costa, who's a distinguished professor of ecology and evolutionary biology at UC Santa Cruz and past director of the Institute for Marine Sciences. He'll introduce Terrie.
Thank you, Eli. It's a great pleasure to introduce Terrie. I didn't realize this was the first Landesman's lecture. Terrie's already, she was the first chair of the first ocean health chair, the Ida Benson Chair in Ocean Health. So she continues to do a whole series of first. Terrie works on, and you'll find out a lot about, Terrie works on sort of ecophysiology, the interface between physiology and ecology. And her work is really how animals work, how they live where they live. And I was just thinking about the fact that one of the keys of Terrie's work is that she works on big things. But it wasn't always that way, because she did her undergraduate and graduate work at Rutgers University. And for her dissertation work, she worked on mink. And she studied amphibious behavior. So her ability to study things in the ocean, which is the other part of Terrie's career that I'm not sure she's going to talk about today. But you might also ask her about the newt study. She did some work with some newts, and I'll leave it to her to tell. So she went from Rutgers to Scripps Institution Oceanography as a NIH-funded postdoc working with Jerry Koyman. She continued, there she took swimming, instead of mink, she looked at swimming and things like harbor seals and California sea lions, and then started working on sea otters. She went to Hub SeaWorld, she went to the San Diego Zoo. While she was at Hub SeaWorld, she worked on effects of oiling and how to clean sea otters which led her to set up the oil spill rehab facility for the 1989 Exxon Valdez oil spill. So Terrie became one of the world authorities on dealing with a catastrophe and trying to figure out how to deal with sea otters that were full of oil. From there, she went to the Navy in Hawaii. She got to spend a fair bit of time in Hawaii, where she then continued studies on swimming and locomotion, and then spent a year at the Office of Naval Research. And in 1994, she came here and as an assistant professor, and she's been here ever since. One of the things I thought was interesting is that Terrie has a quote. She's a child of the 70s, where there's first Earth Day, remember all the firsts, discovered and fell in love with physiology, which how can you not like that? And then Gloria Steinem and the Women's Lib movement said, we could do it all, so she did. So I won't spend a lot more time on this. She's worked all over the place in Africa and Antarctica, in Australia. She's done so much work in the Antarctic that they named a place after her, Terrie Bluff. She decided that Williams Bluff wasn't quite, everybody's got a Williams, so it was Terrie Bluff. And I've actually been able to fly over that and see Terrie Bluff. She's a fellow in the California Academy of Sciences, a fellow in the Society of Marine Mammalogy, fellow in the Zoological Society of San Diego. She's an Explorer's Fellow in the Wings World Quest. Some time ago, she was recognized in Discovery magazine as one of the top women researchers. She's received the Ricketts Memorial Award from the Monterey Bay National Marine Sanctuary. She's been the Bider Lecturer on the August Krogh Lecture for the American Physiological Society. And I'll end with last year, she was the recipient of I have to read this because it's the Daniel Gerard Elliott Medal from the National Academy of Sciences, and it's the award for the evolution of Earth and life. So without further ado, and also, two other things. She's not only done great science, but she's written books. She's got 2 books. One of them won the AAAS Award for Science. It was about a Hawaiian monk seal. That book stimulated a group of students in Texas to to collect plastic and make their own monk seal and to sort of spread the word about monk seals. And then having taught with Terrie, I was always so envious because she's a great artist. I can't draw worth a darn. And when she lectures, and you'll see it today, she really engages the audience. She can do it both verbally and with her pen. And if that all wasn't enough, I'll end by saying she's a triathlete. She's done an Ironman, which is a 2.5-mile swim, 112-mile bike ride, followed by a marathon. Terrie Williams.
I'll just preface that by saying my students call me Terrie Williams, or Dr. Terrie Williams, BA, badass. So I want you guys to all calm. Just forget everything that's going on, because all we're going to talk about tonight are cats. It'll be interesting. First, I want to just say thank you. Thank you for putting this on, and to be the first person to talk. Yeah, I'm a little nervous. It's like hard to live up to all of this. What I'm going to do is show you some science about cats, but I also want to give you the journey, all right? So if there's students that are sort of following what we're doing here, I want them to see the ups and the downs of trying to do the kinds of science that we do and the reasons why we do it. And I felt that that was in the spirit of the Landesman group, and we'll see how it goes. The first thing I'm going to do, because I always run over time, is that there's so many people that are involved in this fielded team. There's a UCSC group, Chris Wilmers, I think I saw in the back. There he is. Was a big part of the instrumentation that we developed. And then we were working in Kenya. I'm working in South Africa now. And then the zoos that we worked with in order to do the calibrations for our instrumentation have been just amazing. Busch Gardens was a part of it, so was Wildlife Safari in very big ways, and some of the wildlife groups. In terms of the funding, we've had, should I talk closer? Okay. Is that good? In terms of, wow, that is loud now. Yeah, you control it. Okay, you control it. In terms of the white lettering here, this is to just give a shout out to the people that helped to fund all of this. And as you can see, it's a lot of philanthropy, and now more so than ever. NSF helped us initially, but we rely on a lot of people. And big shout out to Michael Smith and Nancy Lusk, who funded before, during, and hopefully after what we're doing in Africa. So in terms of the title of this talk, I stole it. And it came from a book my parents made me read as a child because I wanted to get a kitten. And they said, you've got to know something about cats before we allow you to have this little animal. So I sort of borrowed that. But it turned me on to biology. It turned me on to what was happening and gave me an understanding of cats. The picture on the cover pretty much explains what cats are about. So I figured I'm going to use it. In terms of inspiration, though, that same year, this Life magazine came into my parents' house, and it was it. It was the power of photographs of these cats in Africa. You know, as a kid growing up outside of New York City, this was amazing to me to see these animals and moving and hunting. And I said, I am going to study these big cats. This is my life. And I was going to be an explorer. The whole idea was that I was going to go out and explore. And so these are sort of the heroes, you know, Shackleton, Robert Falcon Scott, Captain James Cook, Stanley and Livingstone. These are the people I wanted to emulate. And kudos to my parents for not sort of pointing out the obvious, which is these were all stiff, upper-lipped, white male Brits. And this is what I was aspiring to. Before that, I had told them I was going to grow up to be a dog. So I think that they thought this was an improvement. But I knew back as a kid. So I was just in, you know, early grade school, but I had this vision in my head. The dream was I was going to study big male African lions. That's what my life was going to be about. And, you know, speed up in time. I went through grade school, high school, went to college at Rutgers, got my graduate degree, got a PhD, did a postdoc down at Scripps, did a whole bunch of other things that Dan just mentioned, and then was able to land here in Santa Cruz County in 1994, and this was it. This is cat country. And I was really excited to be here just for that reason. And we're all cat people. We all know what it's like to live with big cats in our backyard. And every time I go to the mailbox, this is the thing that I think about, and I go, is this going to be my last trip? Is this my last, you know, picture vision? Because I'm walking a corgi in cat country. And so all of us, you know, at some level, you've got that concern where these cats are out there. And the thing that I found really interesting, why is it the cats that give us that gut feeling of being preyed upon? You know, if you go back in time, well, let me go back up a little bit. One of the things is how they look at you. You have no doubt in this picture which one is the cat and which one is the deer, the prey. You know, they are just watching you all the time. So it's not, you know, it's not without reason that we're a little bit concerned about the top picture. But it's interesting, you know, when you think about it, that there are wolves, there are coyotes, there are big bears, and there are big cats on the landscape with people. And yet it's the cats that seem to create this conflict. And so I want to talk a little bit about the biology of what cats are about, what felids are about today. And this idea of sort of killer appetites, are they really that kind of a killer, or if we made them into something that really isn't as scary as we think it is? So the first thing I'm going to do is give you the secret to life. So I'll bring that up in just a second. These are my observations. Next, how do big cats eat? And then catabolism. So this is a biochemistry of cats, the inner biology that I believe are making them fierce. And then these big fierce appetites versus humans. So the secret of life. This has taken me a lot of biology to learn. And it is energy in equals energy out. That's all it is. It drives every animal that I've ever worked on. It drives ecology, physiology, etc. Every creature walking around has to accomplish this. They have to accomplish enough energy coming in and not expend so much energy going out. The whole idea is they just have to balance it. There are times that there's going to be an unequal balance, but at least this is what they're aspiring to. This makes the difference in your body composition, whether you're going to be large, whether you're going to be really skinny, whether you are going to grow or your youngsters are going to grow, whether you're going to reproduce. This is evolution in action. And it's predicated on how much energy do you have coming in and what do you have energy for all these biological processes. It dictates who's the keystone predator out there. You can look at all these different animals, and it's how much that big animal eats that's going to make it the keystone. And that keystone is going to drive trophic cascades. You already know about sea otters and urchins and kelp. You have to if you live in Monterey. But the thing is, when I look at this, I don't think of it as an ecological concept per se. What I see is you got big things eating a lot of food, and how much that otter is going to eat is going to dictate what urchins are going to live, and what urchins are eating are going to dictate the kelp forests, and it's the entire glue of ecosystems. So my world is all about food and how much an animal eats, when it eats, and then the repercussions that happen as a result of that. And to me, that's the secret of life. And if any animal does a really good job at figuring this out, it is the felids. So I love this painting that was done by Alan Hunt, where it's just got this pride of lions going after a water buffalo. And this is not unusual to see on an African landscape. You know, we saw it just this last fall. It's amazing to watch it, but take a look at the musculature on these animals and sort of what's going on in terms of the dynamic of predator and prey. And this has been going on since the time of saber-toothed cats. These animals are built to eat, and they're classified. Cats, felids, are classified as what we call a hypercarnivore. And I have to admit, I didn't appreciate that for a long time because it was coined by von Blair-Kahn. No. The woman down in UCLA. Blair, that's who it is. But she had coined this for saber-toothed cats, the fact that you had these animals that were built for incredible carnivory. And I didn't appreciate sort of the biochemistry or what that really meant in terms of what these animals are processing. Hypercarnivores are actually classified as animals that have a diet made up of more than 70% protein, all right? Your protein, you've got carbs and fats and some lipid that's all going in there, and you're processing all of that, and that's what's providing your calories. Think about a cat. A cat, and in actual fact, most of them are 90% eating protein. That's what these cats are meant to do. And that, we're finding out, makes a big difference in terms of how they are preying on animals. That biochemistry is a big deal. So just think hypercarnivore, felid, and protein. But this is the other side of it. The other side is this balance that they want. The thing about cats and any animal that's out there trying to get enough calories in a day is that they want to do it as efficiently as cheaply as possible. The easier it is to get their food and then process it, the more that, or the less that they actually have to hunt, because they haven't expended that much energy. So it's about this balance. They're balancing it with all this protein that's coming in, but that they're also going to be trying to catch the things that they are best built to catch. And, you know, even for the mountain lions in our area, they're best built to catch the prey items that we have here. So the ungulates tend to be the normal thing. The corollary to this, and this is every single animal that I've ever worked with, they're inherently lazy. They want to do things very, very easy and efficiently. And so if something is very hard to catch or eat, they're going to shy away from that and they're going to go for the easy thing, hence my dog. So how do big cats eat? This energy in versus energy out. This is what my lab actually works on primarily. And when you start to think about trying to understand hunting techniques for these big predators, you know, all the older kind of ways of tracking was to go out like George Schaller here, who's a personal hero. And you've got a little field notebook and you're writing things down and you're watching lions and you're going to determine how much are they eating, what are they doing, you know, how long does it take to hunt, what might they catch, and when are they catching it. His book is still out there. Brilliant, brilliant book, the Bible that I work with every day, because he's tried to do the kinds of caloric work that we're doing now. But there were big gaps. You can imagine that as you're out there in the field, you're going to miss a lot of the stuff happening at night. It's just if you light an area, then you've ruined the hunt for these animals. And so as a result, there were things that were missing in this. And sort of the longitudinal, long-term, season-by-season kinds of changes that might happen for an individual is really hard to do when you're doing this with a notebook and trying to track animals, and then you're afraid that you're going to be disturbing all of that behavior that you're trying to study. So when I came here, met up with Chris Wilmers, who also had an interest in studying pumas, obviously, and then Gabe Elkham from engineering. And we had this idea that we would move from the typical, you know, sort of radio collars that you've heard of, where people are out there sort of with a radio antenna and following animals and seeing what they're doing. There was enough technology out there that we felt we could make what we were calling a smart collar. And we were going to add all these pieces to it, try and deploy it on wild big cats, and we were going to get all this information from these collars. And believe it or not, NSF funded us twice to do this, because it was going to be the next generation of wildlife monitoring without having to follow the animals all the time. So we combined miniaturized GPS technology. A very small accelerometer had come out at this time, so it's 3D accelerometer, magnetometer, and that gave us a good idea of where animals were going to be going on a landscape. And then we were going to improve on that. So my job was to calibrate these collars and to start to say, how do we interpret the information that's coming in on these collars, you know, stored on the collars? How do we take that and interpret that into behaviors without seeing the animals at night? Could we use the little squiggles that were coming out from the accelerometers to tell us something about what the animals are actually doing? So you can see behind the cat, this is a great drawing that Chris, I'll give him credit for this, had gotten put together. But you see the short squiggles, that's sort of like an animal that's walking, and then an animal that's running, you've got the big accelerometer traces on there. And so we had to correlate accelerometry with the behaviors. And the reason this works is that animals move in really typical ways. I love studying the biomechanics of humans and athletes and animals, because it's just so, no matter if it's an elephant or a dog, they're all moving in similar ways. If you're walking, it's 1, 2, 3, 4 kind of count for each footfall. for an animal. If an animal is trotting, it's 2 and 2. So it's 1, 2, 1, 2, 1, 2, and that the accelerometer is picking up. And if an animal is galloping, it's going 1, 2, 3, 4, aerial, 1, 2, 3, 4, aerial. So we knew that this biomechanics was happening. We went out to Colorado, where there was a veterinarian that had 3 pumas that she had raised. So they were adults. And she said, come on out, and you can basically put collars on them, put your collar on, and we're now going to calibrate the behaviors. So we had animals running back and forth on a track, and we knew how fast they were moving because we were filming them. We had the collar signals coming from that. We looked at the gait patterns. And then we did everything else. Looked at the accelerometer, or matched the accelerometer to resting and panting and grooming and eating, anything, climbing, anything that could affect the motion of the animal. We recorded with the collar and then had the videotapes that said, every time you see this on their accelerometer trace, you know the animal is sleeping, it's running, etc. Really powerful. So, and it was beautiful. You know, when we get the traces out, here's rest on, let's see if I can do this. Ooh, I can. So here's sort of sitting and eating for an animal, so sort of low-level accelerometry. Important thing is if the animal changes position, so if it's on the ground or if it turns on its side, we know that it's doing that. But here was the big boon. This is what I wanted, and that was we could measure each hit of the foot on the ground. So now I could just count each one of these peaks. I'd know this animal had 60 strides per minute, and I knew the speed of the animal. So now, not only did I know where the animals were going, I knew how fast they were going, I knew if they were running, I knew if they were walking. It was just brilliant. But we wanted to go one step further. And this is the one that nobody wants to do because it's hard and it's a little crazy. I wanted to take this, all that accelerometry, and then say, but what did it really cost the animal to hunt? Can I take that accelerometry, those behaviors, and assign a caloric value to it, that sort of the energy expended by the animal in order to get its prey? Now, if I'm doing this with a human and I've got a treadmill in my lab, you take a treadmill and you put a mask on a human and you run them and you measure its oxygen consumption with an oxygen analyzer. And this is the nuts part of it. It was like, let's do it with a lion. So let's take the pumas, put them on a treadmill. No way were we going to get a mask on them, but we knew that we could put a big metabolic dome over them. So there's a metabolic box. There's a hose that comes out of it, goes to our O2 analyzer, and I was like, let's just see if this will work. I had no idea, but I told NSF I thought it could do it. So let me show you what they did. So this is a training film. Oh my gosh, why is it not working? Oh, I'm sorry, I didn't do it. Well, I hope all of them aren't going to do this, honey. So when we put the cats on the treadmill, if they did everything they could not to walk normally. So the video actually shows you where the tread's moving and the cats would take one foot on each side and leave it. We called it skateboarding. And then they would have two paws moving. If they would ride the treadmill to the back and put their back feet, so the back feet weren't doing anything, and then the front feet would be going. I mean, we had every single variation. Eventually, we got the cats to walk not only on level, but incline, because we wanted up and down motions. But this is where the phrase, they're cats. When I would ask the trainers, why are they doing this? They're cats. And that became the mantra for us. So then, and this is through Chris Wilmer's lab, we started collaring animals in the Santa Cruz Mountains and look at the physiological and behavioral responses of the animals relative to their movement patterns. And I'm going to give you just one track from our animals just to show you sort of the power of what this instrumentation does. So this is a puma that's has a collar on it. I now know, we're tracking it to the evening, or actually this is early morning. So we have an animal coming into this scene. We know it from our GPS track, but remember we have accelerometry happening at the same time. At 2 a.m., it's in the backyards of very expensive houses in the Santa Cruz Mountains. And then it moves across the backyards. Over here, it kills a deer, and we know that from the accelerometry. And then it hangs out, drags the carcass uphill, up at 8 o'clock in the morning, and then is eating and resting up there. And then it comes down in the evening at 6 p.m., and then by 6 a.m., the animals move the area. So that's just the motion part of it. If we go into the information that's happening on the collar, here's the accelerometry over time during that kill. This has compressed all of those movements down. Whoops, sorry. But if you take a look at this, all those footfalls are in here. We've got the behavior, the animal hunting. This spike, killing pounce. That's when the animal basically takes the deer out. And if we blow that up and we take all that accelerometry, pair it with the oxygen consumption that we measured, this is the energy expended by these animals. And this is color-coded by behavior here. This is showing you sort of the speed. If it's red or if it's orange, it's going to be sort of the high-level movements. And then by the time it's blue, it's either resting or cryptic. And so the animal's walking around. Here it's moving a little bit faster, expending more energy, sort of deciding what it's going to do, rests over here, starts moving a little bit faster. It's obviously looking around. And here it goes quiet. So either it's moving really, really slowly, or the animal is resting. We don't know which at this point. And here it is, killing pounce. The pounce is so expensive for the animal, it goes past this red line. That's maximum oxygen consumption for the animal. So it's going into lactic acidosis here, wrestling around with the animal, and then it gets to the point where it's eating it down here. So now we know calories coming in, what it's killed, and the calories that have gone out. And it was remarkable to pull this off the first time. And here's the calculating part, and I did that specifically for our mathematics people here. The precision by which these animals hunt, it was just amazing on this first count. So here's the energetic cost of the pounce, and here's the mass of the prey. The Wilmer's group had gone out, and when they'd see these prey events on the accelerometers, or actually off of the GPS, they could go and try to find the prey item that the animals killed. So that's why we know the mass. And we found that if these animals are killing a fawn, you've got, pretty mild accelerometer trace, and it would get bigger and bigger as the animal got bigger. By the time we got to an adult buck, here's the kind of energy the animal is expending. And it was so precise, the line that we could draw from this, that even if we couldn't find the carcass, or they couldn't find the carcass, we could guesstimate what the mass of what those animals killed simply by extrapolating past this line. So it was a really powerful way to get at how these animals are hunting and what they're willing to expend. So let's move up to big lions. Now we had instrumentation that we could go and look at an animal, a felid, that's not 70 kilograms, but upwards of 200 kilograms. This was the lions that I wanted to, or these were the lions that I really wanted to study. This was a 50-year-old dream at this point, and I was like, we're doing this now that we've got these collars. And this is what was happening to the lion's range in Africa. When I started that dream, there was all these areas that are colored, and especially the red areas are showing you the habitat that the animals are living in. And the gray zones here, sort of the hatched areas, are telling you the sort of past ranges that they had. So we had this sort of circle here that the animals were living in. And then by the time we started this, 2014 or so, this is what it looks like in 2019, sort of these fractured habitats that were out there. And these animals were under a lot of pressure, and it seemed to us that we had a tool that could help people in terms of identifying what were the critical areas for these animals, and maybe to help prevent some of the interactions that were happening with humans. So we had to get a bigger treadmill in order to calibrate the collars. So we had to take the same collars, but make them bigger and also calibrate them, because it doesn't translate from large and small animals. You had to actually do this. So I won't even tell you the number of doors that were slammed in my face saying, I'd like to take your lions and put it on a treadmill. It was just like, no, most zoos are not going to do this. Busch Gardens said, come to Tampa and bring your treadmill. So I did. This was a larger custom-made treadmill that we did, and you'll see that there's a tread missing on this treadmill, and it's because these two girls went in there and ripped up the entire treadmill. I mean, destroyed it, and they thought this was the greatest thing. They're just laughing at us, you know. It's like, yeah, that was really great. What else do you have for us? But they destroyed it. And so we were going back to sort of the drawing board on how we were going to do this. Everybody was game. They're cats. But COVID-19 hit. So we ended up having to stall for a couple of years. And it turns out that the felids were especially susceptible to COVID-19. And there were zoos that lost leopards and tigers and lions, and some of the endangered leopards, as a matter of fact, to COVID. And as a result, there was no zoo that was going to let us come in from the outside to work with their animals. It gave me time to look around for maybe lions that aren't going to rip up my equipment. And I found this. So wildlife safari, like I'm just on the phone calling random zoos with lions and trying to find somebody that would be willing to do this. But I saw an advertisement for wildlife safari in Winston, Oregon, and I go, well, at least it's on the West Coast. And their lions look really friendly. You know, they look like friendly, good lions. And I go, all right, they're used to people. This could be good. And the great thing was that Sarah Hughes, who's in charge of their carnivore group, said, yeah, come on up. Might be fun. And I'm like, all right. So we got a bigger treadmill. This is a treadmill that we actually designed with Anthony Pagano, who was a student of mine, and it was for grizzly bears and for polar bears. And it worked really, really well. And it's at Washington State University Grizzly Facility right now. And so I said, can we borrow this and use it for our lions? They said yes. We moved it next to the lion den in Winston, Oregon. Here's all my O2 equipment. We are in the lion den right here. And on the other side of us, with just a piece of chain link fence, are the lions. And so the trainers were saying, well, we think that you should feed the lions some meat because that means they will get used to you and this will be great. But every day that we came in, you might want to put that up. That's what we got. It was, and you have to listen to that. When that is right next to you, it's bone chilling. And it wasn't that they were upset. They wanted to get on the treadmill because snacks came after treadmill work. So they could care less what we were doing. It was just that they wanted to be the ones, and there were 4 of them that we were working with. But to be that close to an animal that has that kind of response, there were actually 2 responses. My poor grad student, Emily, who's shown here, she was terrified. Every time they roared, she jumped. I did the opposite. I was like, I got nothing. You win. It's like, I couldn't be afraid because if they got loose and they were going to kill me, they had everything. I had nothing. So I couldn't even bother being afraid. So we thought we were going to do lion versus lioness energetics and look at the drive to kill. And the first thing that we did is we just wanted the lions to sit, just chill out, lay down, do whatever, stand up, we don't care, but just like do nothing. And they did. They were totally fine, totally calm in the chamber. We eventually turned the treadmill on and did some training for that, but it worked. But here is what happened with the animals doing nothing. And this completely changed my mind in terms of what these animals are about, what cats are about, because I had never seen this before. I calibrated this thing 100 times. I sent it back to the manufacturer going, what am I doing wrong here? And they said, nothing. These are real numbers. So this is what we saw. Here's the oxygen consumption. Basal metabolic rate means the animals are absolutely asleep on their lowest level. These different colors are different measurements that have been made by other studies. One is a predicted value for a lion based on their body mass. One is from a carnivore line, a little bit higher, because they said in carnivores they should have a higher basal metabolic rate. And one is for a sedated lion. So this is an animal so far out, it's absolutely sedated. This is what the baseline for an African lion is supposed to look like. This is what we were measuring. When an animal is just sitting there doing nothing, this is the metabolic rate for a lion. And if they're standing, it's even higher. This is a 3.5-fold increase in metabolic rate for a lion doing nothing. And it blew my mind. This is like sea otters have this kind of a 2.3-fold increase in metabolism because they live in cold water. Why is it that the lions would have this? And we're still trying to figure it out. And so here's the part of the talk where I'm going to make stuff up. We can call it a hypothesis, but I'm still working through this. But for the sake of this talk, I thought that what I would do is sort of give you my thought processes, what I think is going on with these felids, but it is very, very different from an ursid and from a canid. Like I said, I've never measured anything quite like this, and I think it tells us so much about these cats. When we're working with animals in the wild, or cats in the wild, and you're just watching these lions, there are a couple of things that get me. So this is a photo I took. These animals go from nothing to absolutely spring-loaded attack. It is amazing to watch the reaction of lions. They just have this ability, even working with them in the zoos, but especially in the wild, where it's just like, bang, I'm going to be on you. And this one female had wandered in, the one that's on her back, and this other female that's on top had some cubs, and she just didn't want her there. And she was on her in a second. Didn't kill her. She was just basically giving her, an idea of what she thought of it. But I want you to look at her body also. Look at the muscles and look at how lean these animals are. These are not fat animals. You do see some lions with fat on them, but not a lot. Nothing like we see for like a seal or any of the other animals we might look at. But these are very lean animals. They are, in my estimation, on the edge, and I'm going to tell you why. Felids are obligate carnivores. By that, I mean they have to run on protein, all right? I'll explain that in a second, but just have that in your mind. These are protein eaters. And it makes sense. This is your habitat. This is your food source. And so basically, there's plenty of protein out there for these animals, and that's what they are built to process. But here's what's happened over evolutionary time. Felids have lost the enzymes to process, or some of the enzymes, I shouldn't say all of them. They've lost some of the enzymes to process carbohydrates and fats. They cannot be like a polar bear and go from eating seal blubber and then start eating berries. It's not going to happen. They cannot process plants like a canid. They might pick up plants once in a while. Canids also are not even considered an obligate carnivore. Wolf comes to mind as the closest for high protein, but the felids are beyond that. These proteins, and this is a thing that I realized from exercise physiology, when you're processing carbohydrates or lipids or proteins, proteins only give you half the calories compared to carbohydrates, compared to lipids. What that means is that for a lion to get the same calories that an animal that can eat a varied diet is doing, they have to eat twice as much food. That's what's happening here, and it's because of the biochemistry of these cats. The other thing is that you cannot store protein. So what these cats are doing are processing protein all the time. They have to bring it in, digest it. They've got these big bellies after eating things, and then they're going to catch the next thing. That's what these cats are built to do. If they don't get meals, and this stands for your house cats too, if they don't get meals on times, and the reason they're like knocking you in the head in the morning is because they need protein, they need to process that. It can't be stored. So the only thing left for a cat that doesn't have food available is to start eating the protein on board, which is their muscles. And they very quickly go into a starvation situation. So that's what a felid is about. And so it means that fielded biochemical systems are always turned on. They're processing large amounts of protein all the time. What we discovered is that it costs them metabolically compared to a canid or an ursid. So we are dealing with something very different here than some of the other animals. And it explains so much about cats. So this is just my thoughts about it. But when you think of what I just told you, it explains why free-ranging cats have such a huge impact on birds and on small rodents. There was a meta-analysis done by Loss et al. They actually had so many people come after them on Facebook and everything else because they put this out, and they said that, well, with 84 million owned cats in the US, the free-ranging animals are likely to be the single greatest source of anthropogenic mortality of US birds and mammals. Whoops. Because they are eating 2.4 billion birds per year, and they're eating 12.3 billion small rodents per year. That's what cats need to eat. So, just think about it in terms of the way that they're processing food. This is why I think felid attacks are often lethal. Beyond the fact that they are built to kill very efficiently so that if you are attacked, it's going to be tough to defend yourself. But the red here, compared to bears, here's felids, here's canids, the predatory unprovoked attacks are shown here for the felids. And a lot of these are lethal. A lot of them are against adults. For the canids, the lethality isn't as high. They tend to be unprovoked attacks, but oftentimes it's for children and dogs. It's not quite the same as these felids that are going, large felids is what I'm talking about, going after adult humans. More protein. There's nothing, there's nothing that they're thinking about here other than if you are the convenient thing, you're just protein. This is why I think living with cats can be really, really challenging. And if you're interested in this kind of stuff, these are a couple of books that I had, and it really gives you some insight about predation by lions on humans. For all I know, this is the Savo lions that they had talked about in the movies and such. It may be that building the railroad where they did just scared off all the prey, and it turned out that all the humans lying in a tent was a lot cheaper, energetically thing to go after. I don't know, there's been all kinds of theories there, but to me, it was like, it sort of made sense. In Kruger, it's just people that are crossing into the park, and they think that there are really large numbers of people that have been taken by the lions there, and it's because they just walk right into the animals. And then you've seen some of the attacks that they've talked about for these hikers. The good news part of this, though, to keep you from not hiking and that kind of thing, is that remember, what these animals are trying to do is get energy in versus the energy out. These animals are built to eat very specific things. And so if there are ungulates around, if there are things for them to eat, if they are not like a young male going into a new territory that's starving and he's going to try to do whatever he needs to do, if there aren't extenuating circumstances, they're going to go for the thing that they're built to eat. A human can be a tough thing, and this is why with these signs, and Chris can probably give you more information about what you should do. I just grabbed one of these signs, but they tend to say fight back if you do have an encounter like this. And I could see that, you know, if you become something that is hard to eat, if you become energetically really expensive to try and catch and deal with, you know, it may be worth the cat just saying you're not worth it. So what about big, fierce appetites in humans? This is sort of the last part. These are our cat collars, and I just want to show you one. So this is one of the male collars that we used, and this is the GPS unit with the accelerometer in it. The bottom part is nothing more than a battery pack, but this year we put a GoPro camera in the system. And it really, you talk about giving a person insight about what these cats are doing, to have a camera hanging down underneath and to actually like go on a hunt with the lions. I'm going to show you a couple of videos, but again, it gave me a real different perspective about what these cats were doing on a daily basis. You're free to take a look at this, and I will say one thing, it's got 3 very powerful magnets on it. Do not put your credit cards near it. Do not put anything you care to get demagnetized, because it will. So anyway, lion collars are bigger than cat collars. And we took them and did the same kinds of measurements that we had done for our pumas with the addition of the videos. Where we were working, Kenya north and south, so we were in Laikipia and Masai Mara, and then down in Kruger National Park, which is a massive, massive place, as big as New Jersey or Wales, just to give you a sense of scale. This is work that was done with Chris Wilmers, and this is Steve Akwanga, who worked with us, and Lawrence Frank, who was from UC Berkeley, was also a part of this team. And we were working in the Laikipia area. My part of the project was to work and provide the collars for the big males, and then Chris was there on for a year, I think, weren't you? A long time. He was there on a sabbatical. And so he was doing the work on the females, and then we joined up. And this is sort of the picture for that area. It's low scrubland kind of landscape, and it's got ranches on it, and plenty of fences. And then also, you know, local people that are just trying to make a living in the same area as some of these wild animals. And here's the part of the problem, is that you've got these conflicts where you've got people out there with livestock in an area where you've got these lions trying to make their living, and on top of everything else, with climate change, the water sources are changing also. And so there's an incredible dynamic going on there. And this is a headline just a couple years ago that it's unheard of. You're not allowed to kill lions in Kenya. And the fact that the herders felt compelled that they had to do something, and killing of 11 lions in Kenya is a big deal. So obviously the conflicts are increasing. This is my first lion. I know it's like a cheesy thing, but I'm pointing to a sign that says science on there, and I'm doing science finally. It's like after all these years. There's Lawrence Frank with, like I said, our first animal, Kichaka. And Chris, you weren't there when we did this animal, but it was like, because it was the first one and we had darted it and it was on the ground, I go, how much time do I have before, you know, we have to, you know, move out of the area? And they go, 10, 20 minutes maybe. And I said, I'm going to touch every part of this lion. So it's like I grabbed its tail and its ears. Its ears are really hard relative to a domestic cat. They have all this cartilage. But then, I don't know why, I just face planted in its mane. I was just like, okay, it's like, that's what a lion smells like. But it was so exciting to do that. And the other guys were putting all the equipment away. I don't think they ever saw me do it, but I was like, oh, this is awesome. This gives you an idea of the size of some of these animals. That's my hand, and it's just dwarfed by the guys. That's my foot next to a paw print. Just to show you that they are cats, these were two big males that were traveling with each other. We were trying to dart the animal that's on the right-hand side there. And we did. The dart went in, and the other male got so upset. He's behind the one that's down. He's trying to get him up, and he won't go up. And he pulled out the bloody dart, and we were left going, how much did the other one get? We're not going to dart it a second time. Fortunately, it had gone in, but I thought it was so cool that he identified that was the problem and then pulled it out. But and then my husband was left. He actually came on this one, and he was the one we were saying, watch that other male while we work and put the collar on the one that's down. And he's like, what do I do? Just watch. It's okay. It'll be fine. In the end, these were the 7 males that we darted. So we had Kachaka, Terrie, the Chunga, Davy, Headley, and Mkwangwe, and Jasiri. So it was amazing to watch these animals. We deployed the collars for about a year. This is Lion Tracker that was set up by the Wilmers group. And it shows you sort of the territories for all of our males, and then look at all the females that they were looking at, and sort of the dynamics and overlap in this like hippie area for all of these animals. But there's a struggle on the landscape, and we certainly saw that in our animals. And I'm going to just show you one track here, just to give you a sense of what's going on. In order to get our collars back, they have a drop-off release. So we drop the collar, it's a little radio signal, drops it off, and then we go retrieve them. For some reason, Chunga, we were supposed to pick up his collar. We had darted him initially over here, and he just took off. He took off into the upper territories. We couldn't get a hold of him. I had to fly out, and I just told the guys, let it go, and when you can, Lawrence and group, we're going to go up and try and get the collar. Except the collar stopped working. And what we had done, we had gotten GPS track, and the animal had gone up 1 kilometer into that territory, and at 1830 does a complete reversal and hauls back, and then it ends up at that starred area where it's just staying in one spot. When it was moving, it had 45 strides per minute, so this animal was he was booking it, trying to get out from whatever he had encountered. And then if we sort of zoom in on what happened here, now remember, I haven't seen this animal. I only have the collar, so I'm trying to figure out what's happened based on the accelerometry. So here's the track, or the accelerometer trace from the track that I just showed you over a couple of hour period. The thing to notice is that we have 3 dimensions on our accelerometers, and so it's red on top here. I'm not going to go through the details, but green on the bottom. And what you notice is that it's reversed here. It's now green on top, red on the bottom. And I'm like, and then the trace goes flat. And you're like, is the collar broken or is the animal dead? It could be either one. I don't know. I just have the information. But with this, because of that reverse in color and knowing what the accelerometer is telling me, he was on his side. So there's a good chance that something was very wrong. So we blew up this part, this part of the trace, so the very last parts. And I'm looking at this. You got to remember, I'm at a computer doing this, and I'm like, did I record the last footfalls of this animal? It's like it's boggling my mind. But then I'm looking at it and I go, those aren't footfalls. I could not figure out what was going on. And then I started measuring sort of the time differential between each of these peaks. And I looked at it and I'd seen this before and I was like, oh my gosh, we have recorded his heart rate. Because when he fell, the accelerometer must have been over the carotid. And so just by the shape of the trace, and because I do EKGs on animals, by the shape of the trace and the distance between the peaks, I was like, this is heart rate. So what happened was he had a heart rate of about 40, maintained it for a period of time at the very end. Then we had that. And I talked to a bunch of vets, and they said, well, he's in tachycardia. He's probably having bled out or bleeding out. And so the heart, in defending the body, is going to go into tachycardia, which is what it did. And then finally we got this. And you're like, oh my gosh. But wait for it. Maybe that was it. That was the last beat for Chunga. And we recorded that, and if you don't think that wasn't just soul-crushing for somebody that wanted to do this for 50 years and to have, this be the end of one of these animals. And it turned out that, he'd been up in this territory. We don't know if he was shot or if he was speared, but one way or the other, he had bled out. And actually, the collar had been stolen and buried, and we were able to retrieve it and find out what had happened to him. And it was a retaliatory killing. He was in the wrong place at the wrong time. The tough part about Kenya was that for the males, not so much for the females, but for the males, it was really hard. We lost 4 out of the 7 that we had collared. And it was everything from that animal, Chunga, that had died. Davy was shot. One of the animals, Kachaka, was killed because another lion he was traveling with, they got into a fight. And the accelerometer showed us that the one had killed the other one. And then these two, Jasiri and Terrie, the reason it's yellow is that we actually pulled them out of trouble. They had gotten into fenced areas and were starving and everything, and we got them kick-started, so they went on. But it was tough. It was really tough on the boys there. So we did those studies. We're still doing more in terms of energetics in and out, so we're just getting that data done. But I want to give you just a quick ending here on the Kruger part. Kruger is a completely different situation because you don't have people with cattle on the landscape. So this is truly a park, but the issue is predator-prey disease transmission because they have tuberculosis occurring in the ungulate herds. And the real concern is that, that these lions are potentially going to pick up this tuberculosis. So we were invited to come out, use our collars, and be a part of that. There's our team on the bottom. And the thought is it drought or disease that is going to lead to problems with the big ungulates? There, the situation was really different. We were working with a veterinary group, and it was just incredible being able to work with these lions. We put our collars on, but then they were doing all of the vet work to look at the blood parameters to see if they'd been exposed to tuberculosis. And the whole idea right now is, are they going to be a poorer hunter and expend a lot of calories as a result of disease exposure or not? And then, oh, that's Tracy right there. She's here. She came out with me on the team. It's awesome. The cool things that we're finding is that you can collar one animal in a pride and they're all following the same track. So we actually have 2 animals in each one of these areas. They are focused on water availability. in terms of where these animals go, and that has huge implications for the disease transmission because the ungulates are going to be going to water too. So in Kruger, one of the big questions is, do you try to separate out animals and have more water holes and water accessibility to prevent transmission, or do you let it go? And I just want to show you one of the videos. So remember, we've got a collar. The camera is underneath, so what you're looking at is the chin of the animal here. And I'll tell you, water is a big deal. And we would count these things. They're licking at 100 licks a minute. So it's like when they get to water, they take it down, but they don't drink that often. And I've wondered whether this high protein diet, because when you're processing food, you're also producing water. I wonder if in actual fact, lions and felids in general may not be as water dependent as some other animals because of the way they process food. Have no idea, but the other side of it is there's a thermogenic effect. You know, if you eat a lot of protein, you feel hot. These guys pant a lot. Just to put you on a hunt from some of our videos, if you look way in the distance here, it's a little small, but this is a male who's watching elephants right on the horizon there by the sun and trying to make a decision on whether to go after it or to go after something else. Down here, same animal, is looking around, but it gives you a sense of just the alertness. It's running out there to try and check and not chase animals off. But these are the kinds of things that we're monitoring in terms of the effort and just that hypervigilance that these cats seem to have for long periods of time. Energy out looks like this. Animals getting ready to go on a hunt. The cool thing is that when they're going to hunt, it's so interesting to watch the prides come together. And it's like they're getting, animals positioned for them to actually do the kill. And then they do kill. And I didn't give you the more grisly things. This is just an animal chewing on a bone. But you see how dirty the camera lens is on the bottom? It's because the camera lens ends up in the carcass when they're eating, and it's like the windshield of a bad car accident. It's like, it's just, it's like that, and I just wasn't going to subject you to that. One last thing. So you can get these pictures anywhere on the internet, and I don't know what it is about female lions and cameras, but they got, and our tags. So it's always the females, and we have to get the instruments back in order for us to get the videos and the accelerometers. So this is what we were facing, trying to get the collar from this animal, which we dropped. It's between her front paws. And we're in a vehicle trying to decide what we're going to do here. And she starts chewing on it. Now, if she chews on it, she breaks the wires between the batteries and we won't be able to find the collar. So we're concerned. You can always tell a lion that's getting excited by how much it's twitching its tail. So watch this one in the back. She sees the female with the collar, and she sort of sneaks around. She's checking things out. This is taken by Tracy, I believe. And then the bloody thing steals it. So she's got it over there, and then the next thing that happens is she runs away with it. And we're afraid, one, that she's going to break the wires in it, but we're chasing her all over to get it back before she hits the river and drops it there, because then we'll never find it. But it was the strangest thing. The girls with these collars, whether they were on for a year or on for a short period of time, they just stole them. I don't understand it. So the work that we're doing, just so you understand where we're going, we're working with a large number of groups. We have our group in from UCSC. The Masai Mara group has Craig Packer there, who's an old-time lion biologist. Kalahari Desert, Michael Scanterbury from Ireland is working there, and then my colleague in Kruger, South Africa with disease. All of these people are going to be using the energetic profiles that we've developed for these collars in order to try and answer a whole variety of human and cat problems. They wanted me to say something about, why I did this, or sort of how I did this, and what was something, I don't want to call it inspirational necessarily, but I want to say that it's the power of dreams right now. And so often you hear people say, I think it's Buffett said this, it's not dreams, it's going out and it's doing the hard work and the rest of it. And I don't believe it for a minute. I kept this dream for 50 years, waiting for the right opportunity to do this work. And I think that having that dream gave the vision, just said, one day this is going to happen, and I'm just going to wait until the time is right. It gives you the grit and the persistence when things are not working. Like I said, it was soul crushing to see what happened to those lions in Kenya, but you keep going because in the end you're going to make a difference. And here's the evolution of my dream team. In '67, this is what an expedition was going to look like. And it did for 50 years. I went on expeditions around the world. And I'd say 90-something%, I was the only female. This was 2024. We didn't do this on purpose. It just turned out that the 2 PIs were female, and everybody working with us in terms of all the help and in terms of the students working, it just turned out it was a full female team. And so we took a picture to commemorate it because it just, it was just so monumental. And in terms of why we do these things, I got to tell you that I'm never going to be rich. I'm not going to be a millionaire. This is not the kind of research that you get a Nobel Prize for, but, and I'm never going to be chancellor. It's not going to happen. But, but I can say that I got to work with lions, and that made all the difference. And if we saved a couple of them along the way, then done. That's it. Thank you guys so much.
Thank you, Terrie. That was really awesome. We do have a few minutes for questions. So, if not, that's Jean. Wait a minute. It's coming.
Is there anything special about the body temperature of the predators and lions?
It's funny you ask about body temperature, because the other part of the team, while we were putting in, using the collars, they were putting in temperature monitors. So Andrea Fuller, who's part of that South Africa team, her whole thing is thermoregulation. And one video that I just couldn't fit in here was of lions panting. And I got to tell you, as soon as the external temperature got up to about 40 degrees C, these animals are just panting really, really heavily during the day. It's amazing to watch it get turned on. You couple that with processing food, and I think that there is a temperature effect, but we'll know once they get their instruments back. They haven't gotten them yet. So it's a good question. Yeah.
Ed?
With the collaring and with the accelerometer and all the testing you do, have you ever come across, aside from accidents that occurred that you referred to, do you ever come across a counterexample to protein consumption or anything else you've mentioned with one of the collars and one of the accelerometers?
When you say a counter to
I
protein.
mean, to what you're expecting. I mean, everything you've mentioned relative to protein consumption and other things, there's an expectation of a certain pattern. And my wonder was, is there ever any ever in any of the tests with one of the lines, 2 or whatever number, that's a counterexample to what you're expecting?
Yeah, I hear what you're saying. So I can tell you that the protein part of it didn't get developed until way after all of this happened. So the sequence of events were we were putting collars out on the pumas first. We were doing the treadmill work on the lions. I had gotten these measurements, but I didn't understand them. Go out in the field, get all of the data from there. And so what I've been doing for the last probably, 3 months now is going through and saying, why is this animal different? Why would I see an increase in metabolic rate? And I'll tell you, one of the more fascinating things is to start delving into the veterinary literature that talks about what's nutrition for cats, what's nutrition for canids. So I didn't have an expectation starting out. I thought everything was going to be low. It's going to look like everybody else, and truly thought that somehow I had calibrated the O2 analyzer wrong. And so in time, having solved that, done calibration after calibration with a system that we had already done with bears and everything, so it was the same instrumentation. It wasn't that we expected the protein to be important. It was that I discovered that after the fact, and I went, how did I not know this? It was like, people that own cats know that you have to have certain proteins in the diet for these animals because they can't make it. And so the diets of felids are very different than canids, and there's a lot of literature just like from Purina chows in terms of it. So I like I said, I hear what you're saying, and we're still developing it, but the information about biochemistry came after the fact. I went in thinking, going to be a big dog,
There's a question
over
back.
there.
So that was a fantastic presentation. Really enjoyed it.
It's fun.
It seems like every cat of different species accepted the collars. Was there any evidence of the rebel saying, "No way, I'm getting this off or else"? Or just no acknowledgement that they had one on?
No, it's like, I'm just trying to think who's been resistant on anything that we've done. For the collars, I've never had an animal really just be like, I'm going to scratch this off and take this off and do, even a house cat I've seen, they'll get their paw in there. But I think because of the way that they're designed, and because you couldn't get a paw in there, for our animals, we don't have a problem even with pride mates or something coming in and wanting to chew a collar off because it's unusual. It's just the opposite. Like you saw for those females, when we drop a collar, it's mine. That was mine, and now something is missing. And so they'll take them and run off with them and chew them and everything else. So it becomes ownership. The males, I've heard male lions being described as they're just bumbling fools. And it's just like, they're out there getting themselves in trouble. And the collars didn't make that much of a difference, but they also have the mane in there. And I think, I didn't show you some of the big maned collar stuff because there's so much hair. It's actually hard to interpret. I haven't had that. People that put out collars on canids, it's a different game. And so I know for hunting dogs, and we didn't have problems with hyenas, but with hunting dogs, some of them will have protective kinds of, almost like a bolt that will come out so that the mates won't start chewing them off. But no, I'd say the bigger problem was trying to convince animals to walk on a treadmill. It's like, but food works really well.
So we're running over time, but
One
I
more?
have a question from Paul.
Quick one. Thank you. That was a wonderful talk.
Thank you.
Because of efficiency, do cats hunt equally by day or by night? Or because you'd think that being cool would be help you preserve efficiency, but on the other hand, if you can't see as well, maybe you're going to have to run
Yeah,
in all kinds of... I mean, what is the balance? And is it the same for the mountain lions here as it is in Kenya, or...
I'll let Chris answer mountain lions first because he's got a better beat on that. For the animals in Kruger, what we were seeing is that the animals were doing a lot of the groundwork at dusk. And so you get this sort of dusk-dawn kind of peak in terms of the hunting. And for the dusk stuff, they were sort of getting themselves grouped together, and they were doing those observations. So like I showed you that sunset with the elephants.
Yeah.
They were sort of getting the idea of what they were going to do. We're not able to see that well, even as it's getting darker, but they're going out. We haven't analyzed all of the accelerometer data yet to say, here's all the big pounces. So I can answer it a little bit better. But from the videos, a lot of the action was in that dawn-dusk kind of period, and then they would rest in the middle of the night and then continue in the morning kind of thing.
And just one little coda. All the years I've lived here, I've never seen a mountain lion, except about a year ago I was coming back late at 10 o'clock at night. I know it was 10 because it was the BBC News was on in the car. And there was a mountain lion in my driveway, which then ran ahead in the beam of my car.
Yeah.
But I walk up that thing every night to go to my cabin, and it's dark. Is it going to come back? I mean,
I know.
that's what I want to know. I mean, does that mean it's part of a hunting circuit, or what does that mean?
Go ahead. You need to say something, Chris. Chris is the expert on local cats.
Yeah, the mountain lions here are mostly nocturnal. They're a little more nocturnal where there's people involved. And, they have large home ranges. A male around here might have a home range of 100, 150 square kilometers. So they're mostly interested in catching deer. And if you have deer on your property, they will come through occasionally. But it would be fairly rare that they're there. They generally will avoid places with human activity. So if you're out and about, they're probably going to be away. But then the way most people see them is they come up in a car and they're not expecting it. And you see them in your headlights.
Here's a Chris Wilmer story. You got to hear this, though. So I'm walking up my driveway, and one day there was a mountain lion, and it had been sitting on a branch of the tree, and I didn't notice it going down to get the male, but I came back, and it jumps off, and it goes off into the woods. Same day, he calls up and he goes, "Hey, guess who's in your backyard?" The thing had a collar on, and he knew exactly where that thing was. Thank you, Chris. I didn't die.
Well, thank you, Terrie. I think we're going to have to stop here. Great.